So our class is aquaculture, and we get to do our lab out at the Avila Pier at Avila Beach. Aquaculture is kind of, the farming of marine organisms. We set up our experiment, and that was to kind of track the growth rates and mortality rates for oysters at two different depths of the water column. That we're doing a statistical analysis of the two depths to compare, to see which one is best for the oysters.
We would put all of the cultures, which is basically half an oyster shell, which we would use those as plates, and on each plate we would put what's called spat, and it's basically the larval form of the oysters. Basically, they gave us all the tools, ropes, hoses, baskets, anchors, and we had to figure out how to put it all together. Each week we would monitor their growth. Lots of things could affect how the oyster grows. One of the main things is the light level, because they feed off phytoplankton. The theory is that there's more phytoplankton at the surface of the water where light is more readily available.
And what we did in our group was, to get better data, we attached letters. They were little cubed beads, kind of, and they had little letters on them, and it helped us keep track of which one, which piece of culture was which.
The hands-on experience is definitely made a lot more sense. It's brought the lecture into my hands, which has made it a lot easier to learn.
The whole learn by doing thing, I love that. The experiments that we get to do, those are always stick better, than something that I just learned about in lecture.
This is an ideal classroom for me, definitely. I think anybody would say the same thing too.